For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Season 4, Episode 3 of Surfing the NASH Tsunami, our wrap-up of an amazing and exhilarating NASH-TAG 2023, starts now.
This week on Surfing the NASH Tsunami.
The spirit, the energy, the passion, and really the true collaboration from academia all the way through to industry and policy change makers is what's so special about NASH-TAG.
I'm not going to go forward biopsying my patients anymore because I have a blood-based marker and I have an ultrasound-based marker that I can use. And with that, I'm going to come up with the indication for therapy in the future.
We're going to realize that NASH is actually an easy disease to treat. And I think we started this field on the wrong note, relying on histology, focusing on F3, F4 for a disease that should be treated like dyslipidemia, targets early. And we're going to get to a point of preventive hepatology.
The idea that we can give drug to patients and figure out relatively quickly who is getting benefit and who isn't, I think makes this whole proposition more viable for patients who have to take the drugs and for whoever's paying for them.
It's not as if we want to turn around and say the day after a drug is approved, okay, now start identifying and treating NASH patients. Those patients should be connected to care today.
Last week, over 300 attendees representing the range of fatty liver stakeholders came to Deer Valley, Utah, or joined online for NASH-TAG 2023. Join hepatology researcher and key opinion leader Professor Jörn Schattenberg, pricing and forecasting guru Roger Green, and our guests, hepatology researcher and key opinion leader Dr. Naim Alkhouri, Novo Nordisk Senior Medical Directors and Liver Health Unit Lead Dr. Amy Articolo, and Global Liver Institute founder and CEO Donna Cryer, as they discuss key takeaways from NASH-TAG and next steps, today on the Surfing the NASH Tsunami podcast.
Hi, it's Roger Green, host of the Surfing the NASH Tsunami podcast, and welcome to Episode 3, our NASH-TAG wrap-up episode. Everyone I've spoken with left the meeting energized, upbeat, and believing that with continued hard work and focus, things in Nashville are likely to get much better and much faster than anybody would have predicted, say, a year ago. In the intro to yesterday's episode, I talked about two sets of surprises we encountered in producing the episodes. Well, the good news is that the new mic appears to be fine. The less good news is that challenges around schedule and logistics continue. To get most of the people we recruited into this episode, we had to schedule 2 sessions. And even then, some voices I hoped you would hear, notably Louise and Rachel, were unable to make either one. I'm happy to report that the way we hear it and read the transcript, the final episode you're receiving includes a tremendous amount of high-quality, thought-provoking content, the kinds of things people expect to get from Surfing the NASH Tsunami. The first part of this episode includes Jörn Schattenberg, Naim Alkhouri, Amy Articolo for Novo Nordisk, and me. This was a conversation of virtually unbridled optimism over the dual prospects of drug approvals and adoption of non-invasive testing. The conversation goes in several interesting places, including how the world will change when drugs are on the market and how we may need some new metaphors and conceptual frameworks to transition our vision of good care appropriately. The second section is a one-on-one interview with Donna Cryer and me. The highly practical, laser-focused Ms. Cryer has shifted to what she describes as regulatory science as her focus, the disciplined practice necessary to garner approval for these drugs and, at the same time, anticipate what will change in terms of payer needs, future clinical trial design, and the roles that advocate companies and individuals can play. There's a lot to chew on here, as there always is with Donna. I love this conversation. In fact, I love the entire episode and hope you will as well. After Donna, we'll have a regular business report, and next week we'll be back to our regular weekly schedule. So without further ado, let's start this one.
Today's first conversation features Jörn Schattenberg, Roger Green, Naim Alkhouri, and Amy Articolo.
The adjectives are mine, but they're really the adjectives I've been hearing from everyone I've been talking with. Amazing, exhilarating, energizing. Scott Friedman wound up talking on Saturday about feeling like he was drinking out of a fire hose the whole 2 days. Sorry I wasn't there. Glad I got to catch it on video. Amazing event. We're going to wrap up in 2 pieces. We have conversations today and tomorrow with different people to share impressions. I'll tell you we have tomorrow because this cast of characters keeps changing. But today we're fortunate Jörn is on the other side of the pond, back in Germany, and is able to join us before he goes to sleep for the night. Jörn, like I was saying, you look better and fresher and better lit than usual. You're blaming it on the books behind you, but I just think Overnight Flying agrees with you.
I'm holding up. I had a cup of coffee, some water, and I'm ready to rest a little bit, but still awake enough to to get this episode in with you guys.
You, my friend, are a total trooper. And then sitting next to Jörn on the left on my screen, we have Amy Articolo, who was part of this episode last year as well. Hey, Amy, how are you today?
I'm doing great. Thank you so much, Roger.
Thank you for joining us. And this is the same room you were in last year.
Yeah, nothing's changed here. We moved a few years ago, as you know, and this room is somewhat done. The only thing that maybe an add, and I don't know if you see it behind me, is some of the dog toys to reflect the 3 dogs that we have, which might be different than last year when I had recorded with you. We might have only had 2, but now we have had another addition to our dog family.
Congratulations.
Thank you.
We're holding firm as 4 cats. I have an ongoing debate with my wife about this, who sees all these cat adoptions and goes crazy. But I told her at 1 cat for every 310 square feet of condo, I think we're about maxed out. And then Naim Alkhouri, who is, I guess, back in Phoenix, has been able to join us as well. Hey, Naim, how are you?
I'm good. How are you? I think the reason why Jörn looks so good is because of all the positive energy at NASH-TAG. It was a good meeting, and honestly, we all left inspired and with smiles on our faces. So it was a good meeting.
Yeah, and songs in your heart, I could hear them all the way over in Pennsylvania.
Yeah.
It just looked like a fantastic meeting. So just to get started, even bad meetings have highlights, but good meetings certainly have highlights. So for an icebreaker, let me just ask each of you what your personal highlight of the meeting was.
I guess I'll get started. No, as I said, it was a very positive meeting. Obviously, when you have positive results from a phase 3 program, that adds a lot of energy. And this is what we had with the Resmetirom Madrigal program, the Maestro NASH results. The press release happened in December, but this was the first time we were able to see some of these slides and share the results. So just to recap, it was a NASH resolution in 30% with the high-dose Resmetirom, significantly better than placebo. And then fibrosis regression by 1 stage and no worsening in NASH in 26%, again, significantly better than placebo. This was even better than expected because in the phase 2b study with Resmetirom, there was no clear signal on fibrosis, but also not a surprise because we've seen a lot of the non-invasive test data, including in patients with cirrhosis and reduction in liver stiffness on MRE and FibroScan. And there's a biologic explanation for the improvement in fibrosis, but it was good to see it in a phase 3. So that generated a lot of positive energy for the entire field. And then also the comeback with Intercept and the fact that they replicated the same interim analysis data now with consensus pathology. We have a little bit more granularity also on what subset of patients are responding to obeticholic acid. The response rate is better for patients with F3. So that's also good because in my mind, this is a medicine I always envisioned to be used in patients with more advanced disease just because of the adverse events and just lower tolerability due to pruritus. But we definitely need something for patients with F3. And I felt like there's room for everyone to add to these results. So the results are great, but obviously with response rate between 25 to 30%, all the other biotechs are still in the game. And I think they have now that energy that they need to take their drug to the next level. So I'll stop at this. I mean, many other things happen at the meeting, happy to talk about, but I'll stop here so others can add their take on the meeting.
That was already very comprehensive, Naim.
That was very comprehensive. Thank you. Amy, or you were in highlight of the meeting. One personal moment, Naim did a fantastic job. One personal moment that grabbed you is where you said, wow, this is really great, or I didn't know this, or I know what we can do with this.
Just to add a little bit, we were reminded on how strong academia is involved in this field at this time too. There were a number of presentations around Nimble, Litmus, upcoming consortia, including Goldmine, Nail NIT, where I think in addition to the strong data that we see from the advanced stage clinical trials, there is a strong consortia in academia that support this. And I think it's that combination at this time that really drives a change in the field and makes very optimistic that we get to a point where we, move beyond the biopsy, have validated biomarkers and ones that are linked to outcomes. That was my special take there.
Well, I think both Naim and Jörn have hit the hot ticket, if you will, of what really was captivating about NASH-TAG. So first, I would totally agree. The spirit, the energy, the passion, and really the true collaboration from academia all the way through to industry and policy change makers is so key at this event. It gets lost sometimes in other types of venues, and that's what's so special about NASH-TAG. So kudos to the organizers and all of the participants because you bring it, and it is absolutely heartfelt, if you will. What really struck me as someone representing industry is just the great dynamic discussion around NITs and how to enrich NASH trials, particularly as when I first started a couple of years ago, we were still focused so much on the transition from liver biopsy. And now I think the dialogue has certainly reflected the advancements of the field in looking at how can we work together collectively. So I appreciate both Naim and Jörn's comments about the true collaboration vision and the spirit of doing this together and being able to enrich trials so that we have the right patients and the right evidence to present for the regulatory authorities, how we can change, if you will, or optimize our approaches and protocols on clinical trials so that we can, again, keep patient safety first, always keeping the patient-centric, which is very much in line with Novo Nordisk thinking. And how can we really start to address some of the issues that are currently in the field, particularly around NASH diagnostics and NASH diagnosis, because we know that from Michael Charlton's excellent end of the year review and the discussions that followed, thinking through the different ways to get engagement and rise the urgency of NASH as a disease, which may or may not have a name change soon, but thinking about which NITs make sense, which ones are going to be reflective of diagnosis, prognosis, and potential treatment response, and which ones are going to be used to reflect pharmacodynamic effect. And so to me, it was extremely vibrant discussions. And my takeaway was this is fantastic. We're really making some effective change in the space that's going to impact patients from here till the future, particularly with, let's hope that we will see therapeutics in the near future.
That's great. And I'm going to give an answer that comes from a slightly different place. One of the questions I put on our agenda notes for today was, where's the dark cloud in the sky? I've thought the dark cloud forever was payers. Well, I still do, but not necessarily in a bad way, because the most realistic question payers are going to ask, which is, how do I figure out who this isn't working for before I've paid for 18 months of it, is not only a question payers are going to care about, but frankly, it's a question we all should care about. Nobody wants to give anybody a drug for 18 months that isn't getting you anywhere. So I thought when Inventiva showed up at AASLD with that first adiponectin data, that was really exciting. And I forget who it was who put adiponectin data around a second drug, Stephen talking about ALT. The idea that we can give drug to patients and figure out relatively quickly who is getting benefit and who isn't, I think makes this whole proposition more viable for patients who have to take the drugs and for whoever's paying for them. And I think that's great. Naim, you put up some of Stephen's slides from the breakfast meeting at AASLD about progressors and regressors and zonal analysis with second generation harmonic convergence from histoindex, which Jörn and I actually interviewed Dean Tai for yesterday's episode on this issue. But between that piece and the specificity we're now able to get into with NITs, I think we can do a better job over the next few years of figuring out which drugs go to which patients and how drugs work together better. And if we can do that at the one end and then at the other end start to figure out quickly for whom drugs aren't working, we've made this a really attractive commercial proposition. We've made it a really attractive proposition for patients, and we've gotten this field jump-started. To me, from beginning to end, it all looked good.
Yeah, I think I agree, Roger. I mean, payers, and that was part of the fireside chat where Amy was so part of the steering committee, need to get into the discussion and need to agree to what we generate for evidence that they link this. And I think the response biomarkers are important, or non-response biomarkers. And I know Novo has also been thinking about this. It could be that for different MOAs, we're seeing different markers. So that's also important to be remembered. This is not one size fits all. And it's something that will come out of these combinations. I think most of the phase 3 trials will have an answer for us along that line. I'm not sure you want to add to that.
Yeah, 100% agree. I think that's something we certainly are exploring at Novo Nordisk too, which is not only are we looking to demonstrate the efficacy, safety, tolerance, and mode of administration that will be optimized so that patients are able to benefit. And I think I'd mentioned this in the panel that it's clinical benefit, and I'd love the collaboration with academia and across different industries of how to make that happen. But it's also about clinical utility. And that, Roger, I do think plays very much into the discussions with payers. What will be needed from their optics in terms of demonstrating value as well as the change over time. And that's something too that I felt was discussed at NASH-TAG that maybe we didn't address, in the past so much, and has highlighted quite a bit, particularly around the advancements on non-invasive tests in the sense of, okay, not only are we talking about pharmacodynamic change, but also the delta of change, because that also is going to have value to demonstrate to the payers about why this matters. Why would they invest and why would they look at foundational treatment and beyond, because it will reflect the pathophysiology of the patient, whether they're what would be the expected progression or a more rapid progressor, as was discussed. So certainly I think there's opportunities for these optimized NITs, or however we're going to call them, that is going to, if you will, cascade to the top and will certainly have relevance according to guidelines, implementation, and clinical utility for the field.
I'm listening to you and I'm realizing I have a hope. Without getting into the politics of it, frequently when you use a two-word phrase, the first word is only necessary because because of where you're coming from. So we talk about NITs, but in the context of patient clinical therapy as compared to drug trials, they're just tests because biopsy isn't a realistic option there. So at that point, I'm not thinking about non-invasive tests. I'm just thinking about best tests. It will be a focus shift that we all need to make. Naim, I thought your comment about biopsy being dead in clinic but not in clinical trials, or exactly, I forget exactly how you said it. I thought it was dead on. And if that's the case, then we should just be thinking about tests that happen to be non-invasive.
Yeah, I think this is a very important discussion. And one point I want to highlight is what is futility, right? Because the bar is set too high by the FDA and EMA, requiring histologic endpoints within 12 to 18 months. So futility, we need to be careful about this word, because if people did not respond based on this histologic criteria, but we are seeing improvement in ALT, liver stiffness, other biomarkers, maybe pro-C3, then we cannot say that this is futility. I think futility should be only the patients that progressed in the clinical trials. This is probably not a good medicine to stay on, but there's that category of patients that stayed the same. And we really need to tease out these patients and figure out maybe utilizing things like histoindex, other, AI, machine learning, pathology techniques. Who are the real maybe non-responders versus people that are responding, but we're just not capturing the response on the traditional histologic interpretation that we are utilizing. But this is important because in the context of this disease, you cannot say a medicine is futile after 12 months just because someone didn't reduce fibrosis from F3 to F2, or they didn't achieve that NASH resolution definition, which is so arbitrary. And with the NASH resolution, you can go from an NAFLD activity score of 8 to a NAFLD activity score of 3, but still have one on ballooning, and you are labeled a non-responder. That's not the right way to approach this. So I think we need to come up with the consensus of what futility is in the context of a phase 3 clinical trial based on histology, dig deeper into this. And I agree, it's very important not to stay on the drugs longer than you need, because you also have opportunity to go on other drugs that might be more beneficial to a specific patient. But we also don't want to jump the gun too quickly and maybe lose some beneficial effects of specific drugs just because we didn't capture it on a repeat liver biopsy.
Okay, so first of all, spoiler alert, I've got probably two of the perfect people on the podcast to have as I say this, because I'm going to turn to Naim and Amy for a lot of help on how to get this done. Naim has been after me for a year to bring payers into this podcast. I haven't done it yet because I couldn't figure out exactly how to do that and what to do it for, but I think we're there now. So I'm going to look to Naim and Amy, to you and to other folks I know who are talking to payers about who to bring in and how to bring in and how to move this conversation to that level, because that has to happen. And when you make a really interesting point, you almost describe three responses to therapy. There's a positive response, there's a futile response, and then there's a third response, which is somewhere in the middle, where we don't know yet what the benefit will be, but we can be confident that the things that tell us it's failing haven't happened. I just had that thought for the first time listening to you.
No, I think you're absolutely right. You have these patients that did not progress, but did not regress. They're still in the same category based on the histologic reading, but a lot is happening. And we can get a glance at this from the AI digital pathology, but also with the NITs. So someone that started with F3 and the repeat biopsy is still F3, but then if their liver stiffness went from 14 to 10 and ALT from 70 to 30 and pro-C3 from 20 to 15, that's probably someone who's heading in the right direction despite remaining at F3, as opposed to someone where things are not budging, or maybe potentially even they're getting worse. The stiffness is now 20 from 14. Yeah, I mean, you can still be read as F3 on a biopsy, but two completely different responses in these two patients. So that's why, the biopsy, I mean, of course, it's the endpoint for the regulatory approval, but it's not the end of the story, and it's not really telling you the entire picture. And these NITs, now we have clear evidence that they actually tell you about the prognosis, and we're actually getting to a point that we are thinking about designing trials just based on the existing data with NITs, and I think it's reasonable. Of course, that's the whole purpose of the NAIL-NIT consortium, and this is the question we want to answer in a more scientific way. But I think the data we have today is looking good for FibroScan, MR elastography, potentially CT1, and even changes in FIB4 index. Maybe not FIB4 at one time point, but at two different time points, and looking if they stay in the same category or if there's a change in the category of the FIB4. So yeah, very complex topic, but I think, again, biopsy is not giving you the entire picture. You have to look at everything.
I think excellent points, and brings me back to some of the discussions that were driven around the NITs. And I appreciate your comment, Roger, about tests. because maybe that's the lingo that we should start adopting, getting out of the specialty realm. And when we start approaching others that will be certainly touching this disease, because that's the nomenclature that they're familiar with. But what resonates with me is the power of machine learning and what certainly could hold promise around these non-invasive tests, or tests if we're going to rename, and their impact in driving diagnosis and then eventually treatment. Are they the same type of tests that will be used to reflect the treatment response? I think that still remains to be seen. But stacked tests, or stacked NITs, as we reference them, will certainly be a priority. Some of the research we've done, and certainly just historically in clinical practice, this is something patients also drive, right? That they like the combination of serologic and or imaging tests. So I wholeheartedly agree that's the likely scenario, or the reality over the next 3 to 5 years, in terms of what these tests will look like, how they will be used in clinical practice, and then certainly reflective of treatment modalities. And to your words, Roger, about getting payers in the discussion, I think it is time for them to start looking at, reflecting, and thinking about how this is going to impact their business models and how NASH patients are going to fit into the general, if you will, process, particularly with earlier intervention. And certainly I think that's where these non-invasive tests, or tests, as they're going to be used across the treatment paradigm for patients, is going to be optimized and ultimately reflected within their own budgets.
So, by the way, my favorite expression, because all the time I used to spend in oncology, is liquid biopsy.
Well, that's familiar to me too.
So I thought you might know that one. Yeah, go ahead, Jörn.
Saying it much more simplistic than Naim said it, I just remind us that liver biopsy is an endpoint we use for conditional approval, and it's not the absolute benefit, as Naim highly mentioned. There can be other beneficial things, and the regulators are well aware of that. And the maximum that we've seen this effect was when the Intercept got the complete response letter in the first turnaround, because they said, well, the overall assessment of looking at histological changes and what comes on the safety side and changes in NITs maybe not strong enough to get conditional approval. And I think it will be viewed that way again. It doesn't mean if you have a 10% delta over placebo on liver histology results that only 1 out of 10 patients benefit. It's clearly a different ballpark, and you have to look at safety and other effects that are not measured in liver histology.
It's interesting you say that. So one of the themes I'm taking out of this first 20 minutes of this conversation, and I think it's right, is that what we haven't done yet in language and in focus is take it away from the world that biopsy created to the world that's really patient-centric and outcome-centric. I mean, I use the example of non-invasive tests, but I think you and the one you and Naim just gave is a far better one, which is that, okay, if only 24% of people show a 1-level regression of fibrosis in a given period of time, all that tells us is that 1 out of 4 people got a clear measurable benefit using some very stringent, if conditional criteria. It doesn't tell us very much about the other 76%. And really, therein lies the whole proposition.
I couldn't agree more. I think, there are many analyses that need to take place to give us a more granular idea about what's going on. I agree, Roger. You have people that responded on histology. That's good enough. They're histologic responders. But also the way we report fibrosis regression still includes that no worsening in the NAFLD activity score and any of the components. And I think we need to also see what percentage of patients achieved fibrosis regression regardless of the NAFLD activity score. That's always important to know, because that's still meaningful to me as a hepatologist. I think it will potentially correlate with outcomes regardless of disease activity. I think also we have the people that progressed, and these are clearly non-responders. But then the majority of patients are in this no progression zone. So no improvement, but no worsening. And we need to report on these patients separately with the NITs and try to tease out, again, like who are the responders that we're just not capturing on biopsy. And hopefully use that to monitor patients in clinic, because I can tell you I will not repeat liver biopsies on my patients. This would be unethical in my mind, and honestly not even very helpful, just because we see it in the context of clinical trials. The stories I'll tell you about biopsies, it's very disheartening. But we had, just an example, a patient was screening for a study for F2, F3, and we sent the slides to the central pathology and it came back as F1. So we're like, okay, well, let's try another trial. Maybe we'll be lucky and get it to F2. We sent it to another trial. It came back as F4. And, I mean, the same slide. I don't know what's going on. I mean, maybe this was a one error, but if you don't think this will happen in the community pathology, I think we're being delusional. So we need to be very careful not to overestimate the accuracy, especially of repeat liver biopsies. The expert pathologists hear things like that. I mean, they think like this is just nonsense. How could this happen? But it happens even in the context of clinical trials, so let alone in the community. So how can we do this and be confident? And I think it's very different between screening and PCP, context and endocrine, and then subspecialty care in hepatology clinics, because our goal is really to have high positive predictive value and be confident that whatever we are telling the patient is actually true. And there's a lot of work to be done here. In my mind, like FibroScan, for example, we have things like the FAST and Agile 3, Agile 4. But in my mind, you need combination of two NITs that are completely separate, like ELF and FibroScan measurements, MRE and ProC3. And if everything is showing you the same, then you are confident that, the disease severity is where you think it is. And there's absolutely no need for biopsy. But then the next question is, well, how do you monitor response to treatment? How do you decide on futility with just NITs? It gets a little bit more tricky. But now with the data from Intercept, Madrigal, I think we should be able to answer these questions in a much more educated way.
Naim, nicely worded. And I can report on some of the cases where I had similar experience with liver histology. It's a test that helps us, but it's imperfect, and we need to move beyond. I think that's bottom line.
And certainly from the sponsor side, we see similar discrepancies and inter-read variability that is a bit of a head scratcher. So I really appreciate the clinical perspective of those that are performing the biopsies and seeing the vast differences. And so I can only echo what Naim and Jörn have already so adequately and beautifully said.
Shifting the focus a little bit, I probably talked to Mazen too much, but one of the things I've come to believe strongly in the last couple of months is that for practical purposes, the thing that's going to matter most is true prediction. Not PPV, not NPV, but really true prediction. Because when you get to the question where the line treater or the patient wants to know, am I doing better or am I doing worse? Indeterminate zone really matters tremendously. So if I've got a test that's really, really good at positive, really, really good at negative, but it kicks 50% of the people to the middle, that's not tremendously helpful in everyday life. I mentioned that, Naim, because I'm responding specifically to your comment, which is I've always thought PPV should be the standard, but I'm now thinking it's true predictions should be the standard. And I didn't think about these things going together, but if you think about futile, success, and somewhere in the middle, you could call that positive, negative, and indeterminate. And the somewhere in the middle is the one that's going to bedevil people. So the smaller we can get that, I think the better off we're doing.
Yeah, I couldn't agree more, Roger. I mean, we have data from Gilead, for example, if you're trying to predict the presence of F3, F4, if you're positive on ELF and FibroScan, your PPV is 99%. So you're like almost certain that they have F3, F4. The problem is that gray zone increases from 30% to 70%. So you're stuck with the majority of patients not being able to tell them exactly what's going on. And this is what we've demonstrated with, comparing different scores like the MAST to FAST, for example, that by utilizing MR technology, you decrease that gray zone, and then you can really categorize patients into, okay, you have that disease state or not, and going from 35% to about 10, 15% is a big deal. The problem is that you still have these cases that just baffle me in clinic where the tests are just complete opposite. Just so someone with a liver stiffness of 17.5, and then the ELF is suggesting F0, F1. And where do you go from here? So if I do another test, am I going to believe a high MRE and say, okay, this is it? Or do I need still to do a liver biopsy? These are clinical realities, I guess. But I agree, decreasing that gray zone is very important. And if we can do it with readily available tests without needing sophisticated MR technology, that would be good.
I think the discrepancies of NITs is, of course, worrisome too. And there are some weaknesses to the test, and sometimes it's an operator-dependent aspect. So if you get an overread on transient elastography, it's easier than to actually measure it low, and it could have been a rib or something. So the devil is in the detail here. You have to actually explore each case by itself then. On the opposite, I think it's an absolute strength if you actually get a concordance in response of biomarkers. And I think that's what Naim mentioned earlier. We trust those results where we see the biomarkers moving in the right way, and we're willing to give up on more invasive technologies then. And so I guess the devil's in the detail. Overall, I'm pretty happy with them, I have to say. I base a lot of my clinical decisions today on these NITs, and as Naim, I'm not going to go forward biopsying my patients anymore because I have a blood-based marker and I have a ultrasound-based marker that I can use. And with that, I'm going to come up with the indication for therapy in the future too.
And you're just spot on, and couldn't applaud that, reflecting back on when I was in clinic and just thinking about some of the comments and presentation that the year in review from Michael Charlton, it really resonated with me that a lot of the NASH patients don't necessarily reside within the HepGI community or the specialty community. They're still, based on the research from Equivia and even looking at ICD-10 codes, that they're still with others, and that, if you fall under the umbrella of HCPs that are, for the most part, self-identify as primary care. And so they're looking for a simple way that's guideline evidence-based, that's going to be able to optimize these tests, these non-invasive tests, because they too don't have a lot of faith in the biopsies. They too are going to avoid the biopsies, and that's part of the the, if you will, answer of why there is such an under-reporting, under-diagnosis, undiagnosis of NASH. And so with the advent of these tests and certainly the guidelines, the evidence, the data, it's wonderful to see. It's refreshing to see that we're moving forward and moving past reliance on the biopsy as a means to diagnose and monitor patients. And certainly that's going to, I think, also grow the field.
Amy, I couldn't agree more. One note about that data point from Michael Charlton. If I remember it correctly, the other side of that point was 200,000 ICD-10 reports in what should have been an imputed 16 million case population. So to me, that doesn't say the bulk of patients are sitting with primary care. That's sitting the bulk of patients, we don't even know who the hell they are.
Wholeheartedly agree.
Yeah, we have these moments from time to time. There was a paper presented at EASL in 2020 from someone at Innova who took a look at what indications were on death records and used death records that had NASH listed on them to ask what were people dying from, and said that, well, the cardiovascular is only 9%, and only 9% of the death records that have NASH listed on them have a cardiovascular issue. Well, the problem is Ian Rowe did this work and brought it to podcast. If you work those numbers out, our death rates are only 10% of what they really are. Point being, an awful lot of people are dying of heart disease without NASH on their death record when, in fact, they're evidencing it. And if you think of how death records are created, that makes a lot of sense. This is the same thing. I took a look at that and immediately asked, what about 15.8 million people? You know, by the way, they're all being seen in primary care also, but no one's even thinking about NASH for them.
So the other thing I was going to say, because the word popped up 2 or 3 times already now, guidelines. And actually, AASLD released its NASH guidelines today. It's on the website, and there's already discussions ongoing, but it covers a nice table where you have NITs and you can use them to rule in and rule out certain conditions, cirrhosis, for example. And it really gives us the evidence, or at least the guideline-based evidence now, that we can use those in everyday care. And also, I think it's important for the clinical trials, obviously.
Let me go on to last year in the NASH-TAG wrap-ups. I asked everybody a question, which was, I asked them on a 1 to 6 scale, rate their mood coming out of the meeting from 1 to 6, where 1 was thoroughly depressed and 6 was over the moon. And at that point, the question was about the fireside chat and are we making progress with regulatory? So I want to go back to that question 1 year later and see where do we rate ourselves now and why. And then I want to take a look at it again on the overall, on where you feel the vitality of this market is right now as compared to what was a year ago. Let's do regulatory first. Amy, I know what your score was last year.
You know what?
Because you answered this
Roger, yeah, and I don't remember, so you'll have to tell me what I said. So
yeah, but you've got to go first.
Okay, I will happily go first. So I'm going to say my mood now is 6.
It was 5 last year, folks.
Okay.
She called it, in fact, a solid 5 last year.
All right. Yeah. I am I would say 6+. I mean, that's how invigorated I am about opportunities for us moving forward, particularly with the positive readout of Madrigal and Intercept and their steps moving forward, but also results from the Mosaic meeting and some of the dialogue and opportunities to explore. And the third reason I'm so really excited is because of the opportunities to do this as in a collaborative fashion with the consortium.
Stay on are you 6 on regulatory or 6 on overall or 6 on both?
I'd say 6 on both.
All right. So now, because the world is always changing, Naim, you're clear and Jörn has suddenly become fuzzy. So why don't you go next?
Well, I'm 7 on everything. I'm beyond excited also. I think number one is now we feel confident that these endpoints by the FDA are something that we can achieve in the context of a phase 3 clinical trial. At some point, we're questioning, are we going to ever be able to actually achieve these endpoints and show the regulators that, you know, this is worth conditional approval? So I think we've shown this now with Madrigal. Intercept data is a little bit more reassuring in terms of the safety, although, I mean, I just see the same issues popping up still. We're still talking about the pruritus in about 50% with the higher dose. We're still talking about DILI and potentially direct effect in terms of hepatotoxicity of the medicine. We're still talking about cholecystitis. So a little bit better, but not much, to be honest. But I feel like, number one, we showed that these endpoints are not impossible. Number two is we're going to have a lot of NIT data now from the phase 3 programs to show us what we can use instead of biopsy. And then to see the updates from Litmus and Nimble. Goldmine is enrolling also strongly to show us the value of MRE. Nail NIT is about to start soon. So this is all good. So I feel like all the pieces are falling in place and I'm very happy.
You know, and I normally like to take controversial standpoints, but I can't. I'm with Amy and Naim. I'll put it at 6.5, I guess. I full-heartedly agree. I knew something that maybe wasn't there this time was the regulators, but as Amy said, you know, there was an intense dialogue at Mosaics and there were positive vibes around that. So I don't think you can have the regulators at your table every 4 weeks. So it's not really a downside downside for that one.
Because my first career was in politics, I'm mindful of the idea that when somebody says 10,000%, something really bad's about to happen. So I'm going to stand on 6, because every time I get a number, in part it's my statistical training, and in part it's every time I hear a number over 6, I go back to old things like George McGovern asking how long he would stand behind his running mate after his history of depression was revealed. And he said, I'm 10,000% behind him, and he threw him off the ticket 3 days later. So numbers like 7 scare me. But I'll stand with 6. I'm just realizing it as we're having this conversation, but one of the things that gets me there is the change in the questions. When I first encountered NASH, which was in 2019, the questions were basically about what do we have to do to squeeze more regression out of biopsies? Well, now we're not talking about regression. We're not talking about biopsies. We're not talking about regression being the only thing we're looking for. And we understand a lot more about the liver so that we understand that 2 drugs can get to the same number and maybe hit the same zones and maybe work in completely different ways. And the promise that gives to the vitality of the market. So I'm with you. I think 6 sounds about right.
I mean, no doubt there's going to be some setbacks in terms of negative trial data potentially, but the 6 comes in because it's doable, it's druggable, and we have the tools that have been worked on for so long. So really, the field is set and well prepared for things to come.
I agree with that. The other thing, and Amy, I'd love your feedback on this, particularly commercially, I find attractive, is that the results of those first 2 tests say that we can get a drug over the finish line and that there's an amazing amount of room left for innovation around modes of action, combination therapies, learning how to map the liver better, that people are going to be spoiled for choice about what to do first for years to come, I think.
Roger, you're spot on. And certainly that's Novo Nordisk's aspiration as well. And looking at not what can, some it's just the beginning, right, of, where we can positively impact patients' lives and really thinking about from a patient-centric approach, what diversity of mechanistic approaches are going to help amplify the patient's response, the patient's benefit. And I agree with the comments that Naim and Jörn had also said about it's not necessarily that patients should be started on something and that there should be some sort of like taboo that they failed or they just failed to respond, but more of, okay, that their course of disease at this time had them progressing. And so consideration for additional therapies or reflections on what type of therapies, also reflecting on combination potentials, different mechanistic approaches is certainly very much in line with keeping that patient-centric approach. And certainly I do believe from a commercial perspective makes sense because we know that there's no one size fits all, whether it's a therapeutic, it's a test, not invasive test, or it's the approach of when that patient is diagnosed because of the course of this type of disease. And so having those treatments available, the different tests that will be able to reflect, and certainly making it make sense for payers and those that are going to open the access doors is very much in line with our thinking as well.
One of the other things we're talking more about is bariatric surgery. The Suzanne Sharpton presentation, the Splendor paper keeps popping up again and again, which I think is a good thing. Amy, as a company that manufactures and promotes drugs for obesity, where do you see bariatric surgery playing directly into this, and where does it play into your thinking about development and where the market goes forward?
Yeah, that's a great question. And certainly we welcome in terms of the space of obesity, all modalities that can benefit patients. Certainly with our asset, we're hoping and happy to see that patients are going to benefit. And certainly there could be a role for bariatric surgery either as an and, not an or. And so we certainly have certain independent sponsored studies that KOL such as yourselves have come to us and said, hey, what do you think about these kind of ideas? And certainly it's something of interest in the sense of looking at how can the patients be best managed. We know that certain patients will undergo surgery and then have regain, and there could be a benefit there for the therapeutic or medical intervention. It could be a combination of medical, lifestyle, as well as surgical. And so in terms of patient eligibility, it really depends on the individual patient. And our company certainly is very supportive of getting the patients the best treatments available. And we don't see it as an or, we see it as an and.
I mean, I think bariatric surgery obviously is a great treatment option, but when it's all said and done, give it another 20 years, we're going to look at bariatric surgery the same way we think about drilling a hole in your head to treat headache. It's just too aggressive. And honestly, with drugs like semaglutide, tirzepatide, next generation, the initiation of treatment of obesity will happen way earlier in life. So the minute you start gaining weight because you went to college or, whatever, change in your life happened, you're going to get on these medications. We're not going to let people progress to BMI of 50 and 55. So we're going to treat earlier, and surgery is just too aggressive. I love, my bariatric surgery friends, and I work with them, and it's an effective treatment, but it's really life-altering. It has complications, and the acceptance is low by patients, not because they don't want to get better, because it is complicated, and it's not an easy solution, and it's not what we want to do. And the minute you have an alternative that's less invasive, we will go with it, whether it's bariatric endoscopy or the medications. But I think at the end of the day, the concept is there that if you induce robust weight loss, you're going to improve your NASH. When it's all said and done also, we're all going to realize that NASH is actually an easy disease to treat. And I think we started this field on the wrong note, relying on histology, focusing on F3, F4 for a disease that should be treated like dyslipidemia, target early. We're going to get to a point of preventive hepatology. And at the end of the day, physicians are at two different ends of the spectrum. There are people that are so concerned about cost effectiveness and how to maintain status quo. And there are people that want to extend the lifespan of the human race and make sure people never die from liver disease. I tend to be on the second camp. I'm a very optimistic person, and we need to treat early and just make sure that no one is at cirrhosis early on. Obviously, it comes with a lot of costs, and this, brings so many issues, and we don't have unlimited amount of dollars, but this is aspirational. As we progress, we will get there. It may not be in my lifetime, but this is the goal at the end of the day.
Oh, I'm sure we'll get there. And Roger, you got to make a note. I think Naim deserves a couple of grannies for his statements today. He just hit another home run. I would add that the costs and the availability of bariatric surgery are something that's limiting it. But I like his quote that NASH is actually an easily treatable disease. And I agree, we're just looking at an entity or an outcome that's just difficult to assess for that disease at this time.
Jörn, as I was listening to that, I was A, thinking you're right, he's hitting it out of the park. B, I love that statement. And C, I was asking myself how Louise would feel about the phrase preventive hepatology as compared to it should never get near a hepatologist in the first place. Other than that, Naim, I think we're all standing beside you or behind you and saluting on that one. I think that sounds dead right. With that closing question, one thing that's going to happen, and by the time we, hopefully all of us, come to Utah next year, that isn't obvious.
Well, I think we'll have more data on outcome and NITs that are emerging from the maybe initially retrospective data cohorts, but then also prospective data cohorts that are collected in academia. And one idea Amy brought forward during the Fireside talk was actually to pull things together. And she mentioned that one goes to the regulator by himself. It's not as easy as if when you combine this, and there are certain initiatives that are doing this. And I believe we'll be in a position to actually, across a number of different trials, across a number of different consortia, bring that data to a big data set together and give the evidence to the vision that Naim rightfully is painting here.
So, I absolutely agree with Jörn. I mean, that would be my hope and view of the future, is that the next time we, reconvene and we say, what did we do this past year, that we'll see that there's a much higher NASH awareness or disease state awareness. And part of that's going to be really ensuring that the data is more broadly disseminated, that guidelines are known, and that collectively the regulatory landscape has shifted in the sense of looking at this disease as a whole, and that perhaps I might be saying something a little bit provocative here, but answering the question is, do we have enough evidence to make that change in the guidance around clinical trials?
Well, here's hoping. Naim, one unexpected thing that might happen in the next year.
Well, I think, now with the anticipated, hopefully, FDA approval of the first NASH drug this year, I think there's new energy around also pediatric therapeutics and developing pediatric study plans and trying to come up with the path with the FDA, how to get to that point. I mean, I really hope that it does not include liver biopsies in that population. I think probably starting in adolescents, 12 to 17, and then just extrapolating the NIT data from adult into that patient population will be the way I would prefer to see things progress. I don't think the FDA and EMA are on the same page, and, there's a lot of conversations around this. And the other thing is digital therapeutics, and what I see happening, hopefully once we have a drug approved, is to see, can we enhance the efficacy of any of these drugs with digital therapeutic platform to take that NASH resolution rate from, let's say, 30% to 40 or 45%. So that might be something that we start talking about next year, is maybe combination, but not with different drugs, but actually different interventions in addition to a pharmacologic agent.
That's really interesting. I love both those. You and I started talking about this. We need to get episodes on each of those in the relative future. I can't tell you what this is going to look like, but I think this discussion a year from now will be at least one level deeper than it is right now. In the same sense that one of the things we've done on this episode is we've talked about a couple of things that used to be two-dimensional, three-dimensional, right? Regressive progressors versus regressive progressors and folks in the middle. And what does that mean? I don't know what a 4D view is going to look like or where it's going to land first, but somewhere we will have a 4D view by next year. And I think it's going to come out of the consortia and it's going to be around the tests, carefully said. I've now got my own $20 swear jar for NIT, so they'll be around the tests. I expect that a year from now will be at least one level richer.
Our second conversation is a one-on-one discussion between Donna Cryer and Roger Green.
In the tradition of this entire NASH-TAG episode series, we have not yet had any recording session where everybody we expected has shown up. Today, though, we've brought that to new heights, which is that I'm here with Donna Cryer. Louise is tied up in traffic somewhere outside of London. Jörn had some family issues come up, and the other people who were supposed to be here, well, one of them is ill. So I have the amazing good fortune to have a half an hour to do a one-on-one interview with my friend Donna, which is really might be the highlight of my NASH season. Hey, Donna, how are you today?
I'm doing very well. Thank you.
Yeah, I should tell you that Donna looks better over video than she has in all the time we've been doing video together.
Yes, I promise not to pass out. There are times where I've been fading in the chair, practically on my way out the door to the ER while doing the podcast. This is not that. Maybe I should wear a sign on my back powered by Equinox Gym, personal training, and some other things. But I am very blessed to be coming into a new year in great health and mood. And that was only enhanced by going out to Park City and seeing all my friends in the NASH community.
Yeah, I'm so jealous I wasn't there. Everybody has described that meeting as being really a unique experience, packed room, amazing levels of energy. But what was your personal highlight of the meeting?
I just really wanted this opportunity to say the term Fagan nomogram. That's my new favorite word. I just lit up when it was said because I seldom get to learn new words that just have, that I want to just sneak into cocktail parties and just say randomly, see how I can sort of like goat yoga. I just want to see how I can use them in conversations because I just like the way that those words and sounds fit in my mouth. Now, it's also a fantastic concept in terms of bringing more precision to having conversations and delivering results and then the likelihood to patients. So that is fantastic. But I just love the term. And that's my new 2023 resolution is to just see how many places I can use that term.
We'll check back with you later in the year and see how you did on that.
I'll keep a tally.
Now, by the way, I don't think that was my I wasn't at the meeting, so I didn't get to really have a personal highlight except that I watched it. But I'm right there with you. And you and I chatted a little bit about this beforehand. It came up in yesterday's conversation. I'm sure we're going to circle around to it. The idea that we've got to conceptualize what this disease is and really how we treat it and how we teach about it and all that stuff differently, because a lot of the rules that it takes to get a drug approved don't have a lot of value when you actually have to have doctors treating patients. I thought Fagan nomogram was an amazingly elegant way to show how quickly you can get a doctor and a patient to an answer that goes yes, no. And where maybe so really means maybe so. Not, hey, we've got an indeterminate zone of 45%, but that's okay because our PPV is pretty good. Aside from Fagan nomogram, everyone I've talked to about this meeting talks about the amazing level of optimism and energy. What were the sources of optimism that grabbed you most positively or strongly?
So I too share a lot of optimism. Having good data, good results from the clinical side is the first step. However, I have a concern that doesn't seem to be as widely shared as I would like it to be. That does not make an approval inevitable. So what kept going through my mind as I left the meeting and even as I listened to some of the earlier episodes of this podcast, hope is not a strategy. And so optimism is not an inevitability. So there is a lot of work that still needs to be done in regulatory science, term I hope to familiarize this community with, that needs as much intention and invention and investment of time and resources as the clinical science if we are going to yield the benefit of all the hard work on the research side. We really need to focus on our assignment this year on the regulatory side. And I don't think that everybody got there with me.
So first of all, I'm laughing because the first time this podcast tried to tackle regulatory science was on the Intercept CRL back in 2020. And a good friend of mine, who will go unnamed but might be part of this interview, recommended a regulatory consultant to come on the podcast with us, who made a statement that turned out in hindsight not to have been right and caused a whole bunch of fuss and feathers for a long time, beyond virtually getting Steven yelling at the guy on the podcast. So I know what bad regulatory science is. Now let's talk about what good regulatory science means in 2022. Let's just start with what's good regulatory science.
Good regulatory science and a good relationship between community and regulators are when you hit endpoints that you have agreed upon, then the drug is approved. We didn't have that. And so when I hear conversations around, well, we think FDA or we're encouraged by FDA or they said that if they present us with reasonable data, then that just means, we're all good. And I was like, well, how'd that work out for you last time? And so I will believe it when I see it. I believe that we need to be persistent and consistent in our conversations with FDA. I think they need to be granular. I think they need to be starting to be more conclusive. On our side, we need to be simplifying our message. All the wonderful nuance and caveats that we know and hold in our heads as the field is evolving will only cause chaos and confusion when we come to an AdCom. So we need to start switching our mindsets or recognizing when we need to be presenting information for different use cases and context. And certainly the questions and many of the issues that were raised in our fireside chat did not demonstrate to me that there was an understanding of when we needed to change contexts.
So first of all, I'm going to note that good science by its nature is skeptical. So one of the things that you are being, in fact, is a little more skeptical than some of what we've heard. That might not be the sense in which you meant good regulatory science, but it is one of the things I'm picking up immediately. Second, can you give me one example, give our audience one example, lay out what was said, and then talk about why you think that might not have been good science, good regulatory science?
Well, I was having a conversation or tried to have a conversation about what we need to prepare to go up to approval. I was expecting going into a regulatory fireside chat to be discussing regulatory issues. And the conversation kept turning to, should we do clinical trials in this? And should we do, and there was more on the research and development side versus the regulatory and approval side. And so it's as if I was in the wrong room or was present for the wrong discussion, or they were, one of the two, because what I was prepared to speak on in terms of how we needed to communicate with the Food and Drug Administration, how we needed to make sure that we were prepared for health technology assessments, which would come right on its tailcoats. Also, how we needed to look forward to phase 4, even given the lights of legislation that was passed in the last Congress about diversity requirements, for example. And so I was wanting to have a regulatory conversation as the only JD who sort of snuck up on the panel, I guess. And they were still having clinical trial design conversations. And I don't believe they understood the difference.
A couple thoughts about that. First of all, the regulatory structure might be seen as a bit like an elephant, right? What it feels like depends on where you touch it.
Right.
You weren't only the only JD on the panel, you were also the only person on the panel who had worked in FDA from the inside out.
Yes.
I think to some degree, Amy, by dint of being in a company that's gotten a lot of stuff approved already, understood a lot of that. Scott understood a little bit of it, but he's still trying to figure out what do you have to pitch to the FDA to get your phase 3 accepted, et cetera. And virtually everyone else on that panel touches the FDA through the perspective of what is a good trial, not what is an approval. So I think that's a lot of what you heard in the conversation.
Yes. One of the best questions came from Manal Abdulmalik when she asked about how equipoise would be changed after the hopeful approval of one or more medications and how trials would need to be designed now. And you see Inventiva and others switching and sort of anticipating that shift in equipoise. And why it was a good regulatory question was because we need to go into any AdCom that we have this year with an understanding with the FDA that what they require companies to do in phase 4 is feasible, that to have trials of 5-7 year duration or with multiple biopsies or other types of invasive activities is no longer feasible. It's not really feasible now, but it becomes completely infeasible and unrealistic to do, to the point of endangering investment, endangering the potential success, losing valid, good potential medications if we don't have those conversations now. So that was an excellent regulatory question. There are many fields that I don't know enough to ask a good question from. And it was demonstrated in that room that some people perhaps should not have gone to the mic or shouldn't have been fielding questions questions because they did not know enough about the subject to ask a good question. The fact that they did not know that they did not know is troubling to me, because that means they will not ask for help, or they will not reach out to those who do know what they're doing, so that we can be successful in the assignment for this year, the year of our Lord, 2023.
Yeah, or 57 something or 3,000 something, depending on which Lord you happen to worship, right? You got a few different years.
I would accept that answer as well.
How to describe the year we are in is globally a question of some complexity. Although in the States, we tend to have a singular view on this that even, at least the Jews, if you live here, you've accepted
Fair
it.
enough. And when it comes to Chinese New Year, I want to learn what animal I'm supposed to honor as well. I'm still have to use up all of my Year of the Tiger stamps that I have. So I'm putting them on every bill I have this month so I can clear the way.
Now, back to the question.
Yes.
One of the things that struck me yesterday, I love this turn of a phrase. Scott Friedman has for a while used the phrase straitjacket of biopsy. You and I were talking about that in the pre, and you had kind of I thought, an interesting comment about part of it being it's constraining and part of it being it's psychotic. But let me give you a way we haven't thought about that, or not enough people have thought about it. Going back to Manal's question about equipoise, if we need to shorten the trials, then we probably need to think a little differently about, or the agency even needs to think a little differently about what tests and metrics they're going to use for conditional approvals.
Right.
Right. You know, I think we've all agreed that a one-level regression of fibrosis in a 12 to 18-month period is a really aggressive goal and leaves an awful lot of people in, haven't progressed, haven't regressed, therefore indeterminate, if that's the definition you're using. That's not going to work with payers.
Right.
And if we can't do 5 to 7 year trials anymore, so we can't wait to see what happens, then everyone's going to have to get aligned, I think, on how do we get to an answer we're more confident about sooner.
Well, there's this new thing called real-world evidence, and and we should use it.
I know, and there's that amazing new singer, the one who's like a high school kid in Pennsylvania, Taylor Swift, right?
Yeah. I heard she's pretty good. And so I believe that one of I took a lot of notes. There was so much really wonderful content throughout this really excellently organized meeting. One of the things that I noted was the age of different initiatives and databases, of NASH CRN being 15 years, of some other real world evidence, some other repositories being 12 years, how Litmus and Nimble are now advancing. Target NASH has a lot of natural history data. We have networks of claims and Anthem and others, research networks that are connected to PCORNet. And so I think that we can better mine the data that we already have in terms of, natural history. Once we have drugs on the market, be tracking drug responses. But I think that to constrain the field by biopsy or other needs, when the needs of the patients are so great and are so urgent, does not bear scrutiny, does not survive scrutiny.
Yeah, I think that makes sense. I'll tell you one more thing to add to your data set. I don't know about PathAI because I haven't interviewed them on this, but one of the points that Dean Ty made in the interview that Jörn and I did at the end of episode 2 is that because histo doesn't stay in their stuff, every slide they've ever collected is tagged to a patient and reusable or reanalyzable. So as they learn more about what they can do with their routines, they can back apply those to every patient they've ever gotten. And the more we know about the patient, the more you can back apply it to. So that's just another place to look for trying to amalgamate a data set that you wouldn't necessarily think of. I like your point on that. And I agree with it. I didn't tell you actually my personal highlight of the meeting, and I did wind up answering that question. My personal highlight of the meeting was actually adiponectin. And the reason I say that is, we've talked about this a little bit, is that one of the real challenges ahead of us is going to be no payer is going to put a patient on a drug for 18 months where our definition of success is regression of fibrosis and only 25% hit it. So we're going to, over time, need better definitions. We're also going to need a better, faster, smarter way to figure out who is not succeeding on this medication. Right. So the idea that we've now got two different studies, the Inventiva data from ASLD and the data that was presented at this meeting that showed that two different drugs, adiponectin was predictive of non-response, and then Inventiva data Stephen talked about here that ALT was predictive of non-response, says to me it might not be quite that hard, if we put our minds to it, to identify how to tell doctors and payers and patients, all three, this therapy is not getting to where we need it to. And then change therapy, add therapy, do something else. But know that we're not asking people to throw an awful lot of money and an awful lot of time at something that's not going to get them anything. That was actually my personal moment.
And in a more serious mode, one of my favorite moments of the meeting was Dr. Claude Serlin, who's a radiologist, who asked, how can radiology do more? We often see fatty liver disease on slides, and the answer from the podium to not use the word just a little fatty liver notice. I thought that was a fantastic conversation, and I want us to take advantage, and not just at NASH-TAG, but at various meetings to invite more radiologists. I think that was a wonderful olive branch from that community. And, GLI has participated in this Society of Interventional Radiology meetings in the past, and it's certainly something you want to do in the future. But I thought that type of specialty bridge was a wonderful thing to see, since so many patients have been heartbroken by finding out that there was something on an image years ago, but nobody took responsibility, nobody followed up on it, and there was an opportunity missed to start addressing their NAFLD or NASH at an earlier stage. So that was a highlight to me, to have radiology wanting to step up to NASH, which is our theme of International NASH Day 2023. So I'm excited that radiology wants to step up to NASH.
That's great. Listening to Claude Serlin, one of the thoughts I had is that radiology should become, if you will, should come to play the role that biopsy had been asked to play in the past. Mm-hmm. The most precise way we have to figure out, and frankly, as compared to biopsy, which was an awful lot of false precision, which is why I keep calling it the pyrite standard. But radiology isn't fool's gold. Used correctly, hepatogram you can do an awful lot with. For certain drugs, MRI-PDFF is a great measure. Others maybe not as much for the drug, but there is the phenomenon that it measures well. MRE measures well, more precisely than biopsy does. And as you point out, radiology would like to engage.
Absolutely. The other great moments for me on the patient and public health side were some of the really fantastic public health and prevalence presentations by Dr. Charlton and others that will really help us make this case as we're talking to policymakers and the public and introducing more people to, understanding this disease, this patient population, and the scope of the problem. The challenges and the difficulties of getting accurate surveillance data was shown. The sort of extrapolation or having to go from validated or verified data with codes to a very small percentage, I think 3%, that when we look at what an estimated sort of real number would be, or statements about 5 patients being in endocrinology to every 1 patient that's in hepatology. I think that those are really important parts of how we communicate message points and advocacy fuel as we bring this issue further.
I thought the idea that we had 200,000 ICD-10 codes according to IQVIA and you would have imputed 16 million is pretty breathtaking. Frankly, to me, that was more breathtaking than the idea that the codes were coming from primary care and not hepatology. To me, that was a secondary thought, which is if nobody's looking for it, no one's going to get to a hepatologist.
And I think we have to, and hopefully this is what an FDA-approved drug will do. Create this shift. Right now, I feel as if nobody really wants NASH patients, particularly if you're F2, F3. Primary care doesn't want you because they're overwhelmed and they feel like there's nothing that they can really do and you won't lose weight anyways, which I think is giving a lot of short shrift to patients and is missing an opportunity to have people connected to care, ready and waiting for a drug to come on the market. Endocrinology, I think, is stepping up, but not every patient has diabetes, and there's so few endocrinologists, and so that's not where everybody is not going to be touched or benefit from that. And then hepatologists is like, we don't really want, it's an unnecessary referral. I won't say whose slide that was in, unless you're very sick or have complications or complicated issue, hepatology doesn't really want you. And that may be appropriate given how few hepatologists there actually are. But for the patient, that leaves us with nobody really wants us. And so when we talk about, well, why don't you focus on patient education and identification? And I was like, there is a role for that. But my concern and the reason I'm so focused on getting the physician side right and appropriately cultivated and aware and educated and all of those things and the clinical pathways, simplified, is that for many patients, if they went to their primary care doctor today, that doctor might not know about NASH or might downplay it. Oh, it's just a little fatty liver. It's not really something to worry about, or give misleading or mistaken, it doesn't amount to anything. Come back in 2 years, if come back ever even. So it would do more harm than good to send most patients into doctors' offices today until there is that agreement, that commitment by all of the places that patients would go, that this is something serious and this is what we'll do for you based on characteristics or profile that we can create through non-invasive technologies and as well as family history and things like that. So I think that until we have that commitment, in the same way I talk about having an FDA, a regulatory partner committed to following through on what we've agreed on, until we have a clinical community committed to not turn NAFLD patients away, I really am reluctant to send too many people there.
Here's the dilemma on that one, right? If patients don't show up, clinicians will not be motivated to learn as readily. But if they show up, if too many show up too soon, then you have the problem you just described. By the way, in the U.S., where everybody has EHR, pretty much everybody has EHR, that's a more solvable problem, because you can at some point in time simply, particularly in the health systems, push back into EHR and figure out who was there that we should have looked at that we've missed. Once you leave the U.S., where people don't do ALTs in a standard blood panel, a lot of medical records are still on paper, that becomes infinitely harder everywhere else in the world.
We can give out tape measures and have everybody do waist circumference.
Yeah, I know. You said that before, and I think that's right, but it's not quite the same thing, right? And you certainly can't go backwards. I mean, the phrase they used to use in 2016, thanks Obama, I mean, the medical records were Obamacare. EHR was Obamacare. So that, I think, is a thanks Obama, actually, but a true thanks Obama as compared to the skeptical way people used to use that phrase.
As a GAO appointee to the HIT Policy Committee under President Obama, I know that very well. And I think EHRs and patient information goes a long ways. And I think that, one of the things that I hear very clearly is we should learn the lessons from insert patient community that does it better than us. One of the lessons that we learned from hepatitis C is the power of care cascades and alerts. We see some of the VA system having FIB4s available, the ways that we can have the new updated guidelines coded into EHR systems. And yes, those patients that qualify for at-risk NASH really being made forward to a physician through their EHR to be called in, to be screened, to be treated will by necessary be a big part of this. I think that when I hear the debate between general screening versus at-risk screening, some of the arguments are say because of cost. I don't think it's really it. I think it is more that most physicians, even most nurses, most practitioners, are not yet convinced of NASH and NAFLD. And so it doesn't do us any good to do broader screening where they might have a chance to say, see, we knew it really didn't exist, versus having enriched populations, at least at first, so that the numbers that we know will be so overwhelming that they will be convincing to people who are new to this field and will be quite undeniable that something needs to be done. So I just think we need to be truthful about why we're doing these strategies the way we are.
First of all, cost is a reflection of value, right? So when somebody says something costs too much, what they're really telling you is it doesn't bring enough value. And that can be seven different kinds of skepticism, but we kind of have to work on all of those. That there are therapies that are valued, that this disease is going to get to a bad place for an awful lot of people, and or actually several different bad places. If all we're focusing on is who gets the cirrhosis, as bad as cirrhosis is, we're giving lots of people a chance to poo-poo longer. As if we do, fibrosis has its own issues. In fact, even NAFLD has its own issues, and bringing that into things like cardiovascular and endocrinology. And it's cardiovascular, obviously, that people care about. I agree with that. That makes sense. How do you do that? If you had to pick one initiative that you're either working on or can envision that would move the ball way up the field on that, what would that initiative look like?
Well, I think it's if we could be even more expansive with International NASH Day in the community, if every hospital and every company that's in the space as an employer with all of their employees did events, made it normalized, customary, top of mind. I'm sure that many of these physicians, probably even those at Park City, have of patients of great renown and celebrity within their patient groups, and yet they aren't, connected with advocacy to do PSAs and to make this something that would actually reduce the stigma far more than any name change. And so I think that instead of just thinking that we could shame advocacy into being bigger and better and more effective, we could support it, and everybody can think about what is something that they could do within their sphere of influence to really put this on the map and on the radar. We'll be educating a new Congress is one of the conversations I was having today with Team GLI. We will be doing things on state and regional levels. We are, strategizing with heads of gastroenterology and hepatology about how to be more forward within their institutions so they get the attention and the resources that they need. Had conversations about how to add more liver health navigators from the advanced providers and nurses so that there are those resources within health systems. And I think it takes all those things that we have on our NASH scorecard. There needs to be just a bigger engine behind them than that we currently we possess. And the concern I have and have had consistently over this time, gosh, we created the NASH Council in 2017, I guess 6 years ago at this point. And as much as we put out the message of we need to be prepared this far ahead for this, the tendency of this field to want in real-time action, to only focus on, I'm recruiting for my clinical trial and that's what I want to do, or I'm doing this or whatever, instead of helping to prepare for the future, which is now. It's been disconcerting. We've been working on several things to prepare the patient community for the HTA space and for potentially an advisory committee and the after effects of that for some months. But I feel that it won't be until a week after an AdCom is scheduled. Can you send some patients? Well, yes, but that's because we've been doing this work for months. And so I just want the field to really think about and focus on, are all these offices prepared to have NASH patients? It's not as if we want to turn around and say to them the day after a drug is approved, okay, now start identifying and treating NASH patients. Those patients should be connected to care today so that they will be in a place they would have already been assigned, if they can lose weight, to lose weight. They would have already have, demonstrated and documented, success or failure on that in anticipation of potentially going on drug, or they would have done one of 1,000 different evaluations and be set up for success. And all of that needs to happen now, not the day after a drug is approved.
So Donna, I just felt like your Equinox trainer listening to you to tell me at the same time that I've got to go faster, I've got to bend deeper, I've got to carry more weight, I've got to show more perfect form, and I have to do more reps. Not surprisingly, you and I could talk all day. So I want to try to move this on to a couple of questions so that we can wrap it up eventually. Last year, I asked everybody coming out of NASH-TAG on a 1 to 6 scale, where 1 is thoroughly depressed and 6 is over the moon, where they rated themselves on 2 issues. One is the vitality of the market, and the second is the ease or difficulty of getting a drug to market. Where would you rate yourself right now?
5.
Then the typical marketing research follow-up is why not a 6, why not a 4?
I know the steps that we need to do to be successful. I'm confident in my team's ability to get it done. I am hopeful that we'll have the support necessary to be able to do it.
Is it entirely about the support that you get, or is some of it about your ability to educate people to do what they need to do, even if it's not with you in the field?
Yes, it's more of alignment. It's everybody sort of understanding the assignment and sticking the landing.
Okay, so then what does a 5 mean to you then?
A 5 means to me that we have everything that we would need. If we would do it, we have everything here now. So whereas honestly, we didn't before, NITs are not as involved. There were a lot of different parts that were not as mature as they are now, even if we technically did meet the agreed upon goal. So I think today we really, going into this year, it's really quite undeniable. And yet I know enough and I'm experienced enough to know that that does not make it inevitable.
Got it. All right. That makes good sense. So then the wrap-up question I gave everybody is the one unexpected thing you believe will happen by the time we get to NASH-TAG next year. Can't be drug approval. That's too easy.
I go to sleep at night and I wake up every morning dreaming for my Elizabeth Taylor moment that has happened in AIDS, that somebody will step forward and really help us transform people's acceptance and thinking around this condition.
Is that a dream, a hope, or an expectation?
I'm going to step on faith and call it an expectation.
Good. You and I have been talking about this one for a few years also, and I totally agree with you. I think it's at this point in time, it's in some ways the best thing that can happen. Certainly one of them.
And now for our first business report of Season 4.
Slava Ukraini, Choroim Slava, glory to Ukraine, glory to heroes. As we move into the dead of winter, my heart goes out to those who live with minimal heat and electricity because of the savage and capricious bombing of civilian sites, and particularly the civilian power grid.
This is our first full-length business report in a while.
Because of how the year-end schedule actually rolled out, we had 4 business reports in December, but all were abbreviated. Our schedule for the month was a bit of a moving target, in part because we had a hard time getting connected to Stephen to do the NASH-TAG preview, and then the excellent Resmetirom news hit. Things are more predictable now, despite the changing cast from the NASH-TAG coverage, and we should be humming smoothly along once again within a couple of weeks.
We have some new items planned for 2023.
In In response to listener queries, a couple of things will change this year. Within the next couple of weeks, we expect to post a list of all the conferences we will cover over the course of the entire year. Our Rising Tide series, which looks at the implications of the exceptionally high prevalence of NAFLD and NASH in people living with diabetes and or obesity, will become a monthly series. I'm hoping over the next couple of weeks to have my Rising Tide co-host and friend of the podcast, Ken Kusi, on with me on this podcast to discuss our goals for that series for this year. Why you might want to listen, even if you're an industry exec or clinical trial investigator instead of a frontline treater, and how you can get involved in supporting these outreach activities directly. Over the course of the year, we will make over and expand our website, which seems appropriate or overdue for a year 4 project, and I'll report more on that as we go along.
Listener numbers for December were a mixed story.
On the one hand, our December download numbers were far lower than even a couple of months earlier. End of the year has been a crapshoot for this podcast. Some years, downloads have been great, others weak. This is a weak one. Now, part of that may have been the unpredictability of the December schedule. We're getting the factors that cause this under control. Within the next couple of weeks, you will see us return to our predictable schedule of leading-edge stories, as I promised earlier in this business report. Not all is bad news, though. We cracked the Pod Status 250 top medical podcast in the U.S. market for the first time the last week in December and maintained our place there for several days. We reached number 2 medical podcast in Turkey on New Year's Day and have been in the top 35 medical podcasts and top 200 health and wellness in Turkey ever since. This is the 9th. We've been in the top 200 health and wellness and top 35 medical in South Korea for the entire last week. So we've done pretty well in several countries. Very excited about the U.S. And in 2023, we promise to do better. And with that, I'm off. Thanks to Jake, Magic Mike, Eric, and Steve for everything you do every week. To listeners, thanks to you, you make this all worth doing. The weekend conversations will include what we all consider the best sections of the entire NASH-TAG coverage, all first 3 episodes. And we'll be back Monday with another exciting episode of original content. Until then, stay safe, surf on. We'll see you on the podcast. Bye-bye now.
Have any questions for the surfers? You can send them to surfingnash.com, and we will answer on the podcast or the website.